This video is sponsored by Brilliant. Stick around to the end of the video to learn more. All living things are made of cells. Tiny units of life wrapped in membranes filled with a watery fluid of concentrated chemicals chugging away with microscopic machines with the incredible ability to make more of themselves. Cells are the most basic units of life. They are alive, and the things that are made of them are alive, but the things that make them are not alive. The simplest life forms are just a single cell living on its own, but some complex organisms are made of a few dozen, a few hundred, or even a few million cells. A blue whale's body is estimated to contain over 100 quadrillion cells. But all of us started out as just one cell, and through some truly remarkable processes, those cells grew and divided and differentiated and built the people that we are today. All the cells that we see around us came from previously existing cells in this way. Congratulations! You just learned the basic tenets of cell theory, one of the most important and influential theories in all of biology. But there are a few more parts to it that are pretty significant, so let's get started on learning. Welcome back to BioLogic. In 1665, a British scientist named Robert Hooke put a thin slice of cork under a microscope and was shocked to see what looked like little boxes or maybe honeycomb, and that these tiny compartments together made up the whole plant. He called these weird little cubicles cells because they reminded him of the cells or little rooms that monks stayed in at a monastery, and in all likelihood, he had no idea just how significant that word would become. Not long after this, in 1676, Anton von Leeuwenhoek was developing much more powerful microscopes in Holland. He used them to look closely at things like blood and the plaque on his teeth, which allowed him to see human cells, protists, and bacteria. As far as we know, he was the first human to ever see microorganisms, or as he called them, animalcules. Skipping forward over 150 years to the 1830s, two German biologists were on the verge of a breakthrough. Matthias Schleiden, a botanist, was noticing that every plant that he observed under a microscope was made of cells. And at the same time, Theodor Schwann, a zoologist, was noticing that every animal he observed under a microscope was also made of cells. Together, they developed a new unified theory about cells that's still accepted to this day. We covered the three tenets of it at the beginning. They are, number one, that cells are the most fundamental unit of life. Number two, that all living things are made up of one or more cells. And number three, that cells make more cells through cell division. But in order to really understand what cells are and what they do and where they come from, we'll need to understand the structure of a cell and identify some common cellular components. First of all, there are two major types of cells in the world, prokaryotic and eukaryotic. The term eukaryote comes from Greek words meaning good or true and kernel or commander. This is in reference to the fact that most eukaryotic cells contain a nucleus, which contains most of their DNA. The Greek root word pro means before, and so prokaryote means before the kernel, in reference to the fact that prokaryotic organisms don't have a nucleus. And they don't have a nucleus because they're the descendants from early life forms that came along well before nuclei evolved. Let's take a tour of a eukaryotic cell first, starting at the inside and working our way out. The nucleus is where most of the genetic material within a cell is contained, although there are some genes in other places such as the mitochondria and the chloroplasts if the cell has those. The nucleus is separated from the rest of the cell by a double membrane called a nuclear envelope, which is dotted by tiny openings or pores, each of which is lined with an intricate protein structure called a pore complex, which helps regulate the motion of things like RNA and proteins in and out of the nucleus. The structure of the nucleus is maintained by a network of protein filaments called the nuclear lamina, which lines the inside of the nuclear envelope. And there's even an extra tiny sorta nucleus within the nucleus, a particularly dense area called the nucleolus, where special RNA is produced to make ribosomes, which is our next organelle. Ribosomes are made of ribosomal RNA, or simply rRNA, and a few proteins. Ribosomes are protein-building machines. They read messenger RNA, or mRNA, and then signal to transfer RNA, or tRNA, to bring over amino acids to build a peptide. That's right, RNA reads RNA and tells RNA how to build a protein. And that's not even scratching the surface of how cool protein synthesis is, or all the amazing things that RNA can do, but those are all for other videos. Ribosomes exist in two places within a cell. Free ribosomes float around in the cytoplasm, which is a jelly-like fluid that fills your cells. Just as a heads up, the fluid component of cytoplasm with nothing floating in it is called cytosol, and a lot of textbooks will just throw that word in out of nowhere and say these organelles are floating in cytosol, and that confuses the crap out of a lot of new learners. So keep that in mind and don't be freaked out if somebody tries to switch the vocab up on you. Apart from the free ribosomes, bound ribosomes are attached to the outside of a structure called the endoplasmic reticulum, and that is our next organelle. The endoplasmic reticulum is an extensive network of membranes. Endo means inside, plasmic is referring to the cytoplasm, and reticulum means little net, like the network pattern on a reticulated python. Same root word, same meaning. And it is, in fact, so extensive that the endoplasmic reticulum, or as it's commonly called, the ER, accounts for more than half of the total membrane present in many eukaryotic cells. The ER comes in two varieties, rough and smooth, with the rough type getting its name because it's studded with the ribosomes that we mentioned just a moment ago. The smooth ER performs a variety of functions, depending on the type of cell that it's in. These functions include the production of lipids, metabolism of carbohydrates, ion storage, or detoxification of drugs and poisons. The rough ER, on the other hand, helps produce more membranes, because those are always fun, as well as proteins, which are packaged into transport vesicles and shipped off to our next organelle, the Golgi Apparatus. The Golgi Apparatus acts as a shipping and receiving center for the cell. This is where proteins and other things go to be sorted, modified, and sent off to other places around the cell or around the whole body. Things come in and out of the Golgi apparatus by way of vesicles, tiny membrane bubbles, or vacuoles, big membrane bubbles. The Golgi apparatus itself is comprised of a stack of several, sometimes hundreds, of membranous sacs called cisternae, and the whole structure bears a distinctive directionality. The broad side of the apparatus, which is pointed towards the nucleus and is used for receiving, is called the cis face. The curved side, which is angled towards the cell membrane and is used for shipping, is called the trans face. Now, those vacuoles that I mentioned also have a variety of functions, depending on when and how they're made and what they contain. I'll give you a couple of examples. When a cell eats something, in a process called phagocytosis, from the word phage, meaning to eat, and cyto, meaning cell, a food vacuole is formed, as the membrane of the cell just folds in on itself. By the way, the word for something folding in on itself like that is invagination, and that's one of the more fun words to sprinkle into a conversation to see who is and isn't a biologist by who can keep a straight face. Anyway, the food vacuole will then fuse with another little bubble called a lysosome, which is full of digestive enzymes which will break down that food. Notice the word lyse, which means to break something down, and the word some, which means body. So a body or a thing that breaks stuff down. Lots of things in biology that have set boundaries and a specific function are going to be called a something some. Like, for example, chromosomes, which means colorful body. They got that name because they're very easy to stain, and that's how we discovered them. And then there's peroxisomes, which contain and sequester oxidative reactions so that they don't hurt other parts of the cell. They do a bunch of other stuff too. We'll talk about it later. Other cool examples of vacuoles are the central vacuoles that plant cells use to keep water inside, and the contractile vacuoles that protists use to pump excess water out. When talking about organelles like the endoplasmic reticulum, the Golgi apparatus, the nuclear envelope, the vesicles, the vacuoles, and so on, it's important to remember that they're all part of what's called the endomembrane system, a system of interconnected and interdependent membranes that collectively share a ton of responsibilities throughout your cells. These responsibilities include protein synthesis, waste management, metabolism, detoxification, transportation, you name it. And that's all at the subcellular level. But when we get up to looking at the whole organism, you'll start seeing how different organ systems work together in a way that's very similar to how the endomembrane system works. But we're not done yet, because we have a few organelles left to go, and I have less and less to say about them because they are increasingly deserving of their own independent video. We're going to start with mitochondria and chloroplasts. Mitochondria are found in almost all eukaryotic cells, including those of animals, plants, fungi, and protists. Some single-celled organisms have just one mitochondrion, while your muscle cells have several thousand each. Your mitochondria contain a large inner membrane with several folds that we call cristae, all of which is surrounded by a smooth outer membrane. The space between the cristae is called the mitochondrial matrix, and that's where the mitochondrion keeps its DNA. Chloroplasts also have an inner and outer membrane, although both of them are smooth, and within the inner membrane they have a totally different membrane system comprised of stacks of flattened sacs. The stacks are called grana, the sacs are called thylakoids, and the fluid which surrounds the thylakoids is called stroma. And that is where you'll find the chloroplast DNA. We'll talk more about mitochondria and chloroplasts when we talk about energy production. But for right now, just remember that they have their own DNA, their DNA is circular like a bacteria's, and they reproduce by themselves. This is because mitochondria and chloroplasts used to be their own free living organisms, until that is about 1.5 billion years ago when they were engulfed by a eukaryotic cell, not digested, and have been living inside of us ever since, trading energy production for protection. That's called endosymbiont theory. I already covered it in episode 4 of The Light of Evolution, and I have a strong suspicion that we'll be talking about it a little bit more before this series is done. The cytoskeleton is a network of microtubules, microfilaments, and intermediate filaments that provide a support structure for your cells the same way that the nuclear lamina provides a support structure for your nuclei. And depending on what part of the cytoskeleton you're talking about, it also plays an important role in changing the shape of the cell, moving organelles around within the cell, or just moving the whole cell. We're going to talk about the cytoskeleton a lot over the course of this series, especially when we talk about things like muscles and cilia and flagella, because there is a lot there. But for now, all you need to know is that these filaments and tubules are like the thick but flexible cables that hold up a big structure. And lastly, on our way out of the cell, all cells are wrapped in a flexible and semi-permeable cell membrane. Semi-permeable just means that it only lets some things in and out. And some cells are also encased in a rigid cell wall. And yes, both of those things are incredibly complex and they require their own video to explain, so we're just going to leave that there. Finally, let's move on from eukaryotic cells to prokaryotic cells, which, as I mentioned earlier, don't have a nucleus, nor do they have any membrane-bound organelles. Instead, their DNA is concentrated in a region called the nucleoid. They sometimes have extra DNA in the form of little rings called plasmids that just do other stuff, and the rest of their cell is just full of ribosomes bumping around, floating throughout the cytoplasm like they own the place. It's a mess. But don't let the relative simplicity of prokaryotes fool you. Some of them have really cool features, like flagella that they use to propel themselves, or cilia that they use to crawl and eat, or fimbriae that they use to attach themselves to other surfaces. They also have a cell wall outside of their cell membrane, and some of them also have a jelly-like outer coating called a capsule. And that's something that makes some bacteria so dangerous, because that capsule protects them from a lot of things, including antibiotics. Last but not least, let's take a look at the life cycle of a cell, otherwise known as the cell cycle. About 90% of the cell cycle is what's called interphase, where a cell duplicates its DNA and grows in size. Interphase takes place over the course of 3 subphases. G1, or gap 1, is when the cell prepares to synthesize new DNA. S phase is when the cell synthesizes new DNA. And then G2, or gap 2, is where the cell prepares for mitotic division. Mitosis, or M phase, is a relatively very short and very dramatic stage of the cell cycle, and it is broken down into 5 stages. Prophase is when the chromosomes fully condense down into sister chromatids, and the centrosomes move to opposite ends of the cell to begin to form thick filaments called the mitotic spindle. Prometaphase is when the nuclear envelope fragments, and the mitotic spindles begin connecting to the centers of the chromosomes at a site called the connectochore. Metaphase is when all the chromosomes line up along the equator of the cell. Anaphase is when the sister chromatids separate and begin being pulled to opposite poles of the cell. Finally, telophase is when the sister chromatids reach the opposite poles and new nuclear envelopes begin to form around them. The formation of new nuclei marks the end of mitosis, and the cell will then begin dividing in 2 through a process called cytokinesis, where the cytoplasm is divided into 2 parts by the contraction of a ring of actin and myosin filaments around the circumference of the cell until one is finally pinched into 2. Gross. So now you know a little bit more about cells and how they work. Their story is a rich one that shows how scientific discoveries are made, how they build on one another, and how we've come so far thanks to the dedication of a handful of particularly curious humans. Just think about it. Hooke used his microscope to identify cells back in 1665. Then in 1674, Leeuwenhoek saw protozoa, and 9 years later, in 1683, he saw bacteria. In 1833, Brown first saw the nuclei of orchid cells. Then Schleiden and Schwann proposed cell theory in 1839. From there, Kolliker first described mitochondria in muscle cells in 1853. In 1879, Fleming tracked the behavior of chromosomes during mitosis. In 1881, thanks to developments in staining by Cajal, we learned about the structure of nerve cells. The Golgi Apparatus was first described by Golgi himself in 1898 after he stained cells with silver nitrate to see new structures. In 1902, 23 years after Fleming had first described the movement of chromosomes, Boveri finally observed them moving during sexual reproduction, finally linking chromosomes to heredity. In 1952, Palladay, Porter, and Sjøstrand developed methods in electron microscopy, which allowed them to see extracellular structures for the first time. And using their techniques, Huxley provided the first evidence of the cytoskeleton by showing protein filaments within muscle cells. The electron microscope came in handy again in 1957, as Robertson used it to show the bilayer structure of the cell membrane. Then in 1960, Kendrew used X-ray crystallography to describe the first detailed protein structure. In 1965, Dedieu separated peroxisomes, lysosomes, and mitochondria, showing them all individually for the first time. And in 1974, Lazarides and Weber stained the cytoskeleton with fluorescent antibodies, finally revealing its structure. In 1994, Schalfie used fluorescence to track the movement of proteins in living cells. And by the 2000s, Betzig, Hell, and Moriner had developed super-resolution fluorescence microscopy techniques that allowed us to see molecules too small to even be seen by conventional microscopes. And now, in 2024, we need new scientists to explore new horizons and show us new things that we've never seen before. One of those scientists might just be you. We've got a lot left to cover in this series. Even just in this episode, I had to leave a ton of stuff out, so I hope you'll stick around and keep learning cool stuff with me. But until then, I'm Forrest Valkai. Thank you so much for watching, liking, commenting, subscribing, and all the other stuff you do here on YouTube. Have an awesome rest of your day, and never stop learning. Bye-bye! And now, a word from our sponsor. Brilliant is an amazing educational app that allows you to explore science, math, data analysis, programming, and more through thousands of interactive lessons designed by a team of award-winning educators, researchers, and professionals from places like MIT, Caltech, and Duke, all right in the palm of your hand. And the best part is, they're offering my subscribers a chance to try everything that they have to offer absolutely free for a full 30 days, plus 20% off an annual subscription to Brilliant Premium, and all you gotta do to get it is go to brilliant.org/forrestvalkai, or just use the link in the description below. Brilliant's fun and interactive lessons stretch your brain, develop your problem-solving skills, and foster a daily learning habit. As for me, I'm always going to recommend their scientific thinking course. It's a great way to get to know the beautifully intricate science underlying the mundane world around you. No matter what your educational goals are, starting with that course is a great way to ensure that you start off on the right foot. So head on over to brilliant.org/forrestvalkai, or just use the link in the description to explore everything that Brilliant has to offer for free for a full 30 days and enjoy 20% off your annual premium subscription. Thanks so much to Brilliant for sponsoring this video. Thanks so much to you for watching it, and I'll see you next time.